And Phyllis, I posted your email and your WhatsApp number. Can you see that in the chat? Okay, great. Okay, can't hear you.
Yeah,
I saw that you posted it. Let me check it in the chat.
Okay.
Yeah.
Got it. Okay, so everyone could see that. Okay, awesome. Good afternoon. There's Dave. Good afternoon.
Sally, before we have everybody on, I do want to say special thanks, or I guess I should say after we have everyone on.
Yeah.
All your efforts and work in coordinating this, and you've just been such a great leader. I won't say partner because I'm more leader, and we all very much appreciate it.
Oh, no. This has been really a very successful endeavor. Can you let the folks in? Because I've got the main screen up, so I can't do that. They won't allow me to have that functionality. Thank you, Phil.
And I'll keep track of admitting folks, so don't worry about it. Don't sweat it.
Yeah, thank you.
Sally, I do see someone on the phone that is not muted. If everyone could please mute so that we won't get any interruptions, that would be great.
Yeah, I did get an email from one of the participants that they were going to be in the field, so calling on the phone, so it might be that participant, yeah.
I was able to mute them.
Okay, great. Thank you. Does it seem to have slowed down in terms of the folks getting in? Okay, why don't we go ahead and get started, and maybe I'll stop sharing and then we can come on here. All right. Okay. So thank you to all of the participants that have joined today. And if we get further participants, we will just continue to have them join. We're ready to start now. And again, welcome and good afternoon to our esteemed colleagues from Israel. And good morning to our EPA staff. My name is Sally Gutierrez, and I'm from US EPA's Office of Research and Development. This is the fifth and final webinar of the series. And I wanted to offer a special thanks for all of the participants that have joined for all the previous webinars. So thank you for sticking with us, if you will. Today's presentation is a bit different from the previous ones in that it speaks to the research arm of US EPA. And so it is not directly about the regulation and policy and the Clean Water Act so much. It's more future facing. In terms of meeting logistics, again, please mute your Teams if you're on Teams by clicking the microphone icon if you're not speaking. And we are recording the presentation, and all of the previous presentations are now available on the POSI Global website. Please feel free to ask questions in English in the chat. And should you have any questions in Hebrew, please send them to the email address Phyllis@POSI and the WhatsApp number, and we have that in the chat. And we will allow for the translation, and then Phyllis and I will monitor the questions for the speaker. And with that as an intro, I'd like to introduce my boss, my supervisor, Dr. Greg Sales, who is the director of the Center for Environmental Solutions and Emergency Response within US EPA's Office of Research and Development. He's a chemical engineer and has been in the Office of Research and Development for many years. He is originally from California, and Dr. Sales will provide the introductory remarks. So thank you, Greg, and please go ahead.
All right, Sally, thank you. Can you hear me okay?
Mm-hmm. Yeah.
Okay, great. Well, let me add my good afternoons to our Israeli friends and good morning to our folks in the States. Thanks for the opportunity to provide these remarks as we conclude this webinar series with this presentation on EPA's water-related research. Perhaps it is fitting to end this webinar series about future challenges to water quality protection and the ongoing research that will help the agency with its programmatic and policy objectives in the short and long term. So I would like to take a few moments to talk about EPA's research arm, our Office of Research and Development. When EPA was established more than 50 years ago, it was recognized that the agency needed a research arm to make sure that the agency has the best science possible to underpin its regulatory and policy making responsibilities. This strategic construct has proved incredibly important through this time and has enabled the agency to draw on the latest science to guide its work. The research also allows the flexibility to help the agency prepare for future challenges that may not be front and center at the moment. As perhaps you all appreciate, few nations have embedded a significant research organization within their environmental protection agencies or ministries as EPA has. Having such a research institute in-house accomplishes several key things. Our EPA decision makers can seek applied, focused research that will lead to helping make sound decisions in the years ahead. Another thing is that EPA, state and local governments, and our tribal nations can seek our researchers' technical expertise, providing an in-house consulting function to help them with short-term scientific challenges. So for example, our scientists help with local drinking water corrosion issues, or help identify options for cleaning up, revitalizing contaminated urban sites, etc. And finally, the close relationship between EPA decision makers and researchers helps ensure that the research that we do is well aligned with the practical problems that the nation faces and is relevant and impactful. So throughout the history of EPA, the Office of Research and Development has taken many forms, but more importantly, it continues to provide a range of support from analytical methods development for emerging contaminants, risk assessment for the evaluation of health and ecological effects, both microbial and chemical, as well as technology assessment, evaluation, and other risk mitigation measures. This research is conducted across all media, air, water, land, and waste streams. A newer area of research and technical support that emerged after our 9/11 attacks is EPA's mission to help respond to Homeland Security incidents, and we conduct research in that area as well. So EPA has typically invested about 10% of its resources in supporting the Office of Research and Development, a significant amount given the broad statutory environmental protection mission that must be carried out. Of EPA's 15,000 employees, the Office of Research and Development has about 1,500 working scientists and support staff across the country. We organized into 4 centers. As Sally introduced, I lead the Center for Environmental Solutions and Emergency Response. The other parts of the research mission are spread over 3 other centers. One is the Center for Computational Toxicology and Exposure. Another is the Center for Environmental Measurement and Modeling. And finally, the 4th center is our Center for Public Health and Environmental Assessment. And you can get a sense of the high-level areas of emphasis from those names. These 4 centers and other support offices are operated in 12 facilities around our country. The major research facilities, laboratories, et cetera, are located in our states of North Carolina and our state of Ohio. The Ohio lab in Cincinnati is where my organization, acronym CSER, has its headquarters. Most of ORD's applied research on all aspects of water take place in Cincinnati. In fact, our laboratory here is a direct descendant of the United States' first federal water research lab that was established on the Ohio River in Cincinnati in 1912. So a long legacy there. ORD as a whole has a broad research portfolio aligned with major EPA mission areas. We have 6 national research programs that fund the centers. The names of the research programs indicate sort of what areas they focus on. And you'll see it's a broad mission of research. First one is Air, Climate, and Energy program. Then we have the Chemical Safety for Sustainability program. We have a program called Health and Environmental Risk Assessment. We have the Homeland Security Research Program, the Safe and Healthy Communities Program, and finally the Safe and Sustainable Water Resources Program, which we're going to be hearing about some of the research from that program today. Each research program develops a 4-year research plan to guide our investments, and we're in the process of updating our plans for the next cycle. Priorities for this research are set through a collaborative process with our EPA programs and regional offices and external stakeholders such as states, locals, again, our tribal nations. Additionally, the plans and programs are reviewed by our external advisory committee. So given our somewhat modest funding, it's important that we get our priorities right. At the same time, we realize that we have the flexibility to support immediate program needs. As an example, during the COVID-19 pandemic, which is ongoing, as you all know, the Office of Research and Development has been at the forefront of research and technical assistance on topics such as disinfection of surfaces and aerosols, and in wastewater surveillance. The research you'll hear about today, again, is supported by the Safe and Sustainable Water Resources Research Program. This program is organized into 3 bins. First is watersheds, the next is nutrients and harmful algal blooms, and finally, water treatment and infrastructure. So let me wrap up. ORD has many collaborations with international partners, as you know, including Israel, and we value these relationships greatly and look forward to working together on common issues in the years ahead, such as on today's topic of water reuse. Again, welcome to today's engagement. This discussion should be very, very interesting and very useful. So with that, again, let's have a great session, and let me hand it back to Sally to get our main event going.
Okay, all right. Well, thank you very much, Greg, for that really excellent overview of Office of Research and Development. Our speaker today is Dr. Jay Garland, who is one of 2 associate directors within CSER, and he came to EPA about 10 years ago from NASA. Jay has been a real leader in water reuse, antibiotic resistance, climate change, and other topics. He holds a PhD in environmental science from the University of Virginia. So with that, take it away, Jay.
Thank you, Sally. I'm really glad to be here today. Thank you for taking the time to listen to me today. As I call up my presentation here, I always have to chuckle when I hear folks like Greg talk about, long-term versus short-term research. All this seems like very short-term research to me, because the background that Sally mentioned, I worked with NASA. We were working on developing life support systems for Mars missions and lunar bases, trying to develop closed loop life support systems. So that's definitely far term, right? So when I came back to Earth and worked for EPA about 10 years ago, everything seems like it's near term to me. So I should probably stop talking and concentrate on getting my presentation up. We want to go with this view. Can everybody see that? Can they see my slides and my talking head?
Yes, both.
Okay. All right. So
There
what I want to talk about today is
Jay, there seems to be a little animation panel that seems to be up. I don't know if you can close that part to make it look larger.
Or just presentation mode.
Yeah, or just presentation mode. Yeah. If you go down to the bottom there and just.
Well, I don't know if it's letting me control that. Let me go back out of this view. Just 1 second. Interesting. Not sure why it's not letting me change anything.
You might have to come out and go back in. Yeah. Sure. And then. There you go. Now that's it. Thank you.
You're welcome. All right. So what I want to talk today about is as Greg mentioned, the water infrastructure component of the Safe and Sustainable Water Resources Research Program, in particular talking about looking at alternative water infrastructures to and research related to that to inform, what I'm kind of collectively calling a dynamic future here. That's partly driven by climate change, partly driven by aging infrastructure, and in general, the need to kind of rethink how we've structured our water systems. Now, I feel like there's an American saying about preaching to the choir, right? Where you're really already talking to the converted. And I feel that way this morning a little bit because you all in Israel are true leaders in the area of water reuse. But hopefully from this, you'll get a better perspective of where EPA is on this and maybe the states more in general, because we do represent a much more diverse and geographically broad kind of composition, and I think the research and the solutions here are going to be distinctive because of that. Now, if I could just, there we go. So I just want to give you a little bit of background, and I know, like I said, since you guys really are very well versed in this, I want to keep the background to a minimum and really focus on the work that we've been doing, which really has been largely a collaboration with state and local utilities. In a forum called the Blue Ribbon Commission for On-Site Non-Potable Water Systems. This has been an effort that's been in development over the last 6 or 7 years, led by Paula Kehoe from the San Francisco Public Utility Commission, if you have run across her before. And as Greg said, there's a range. We do work to inform, research to inform our program offices as well as our regional and local partners. This is work that's really directed towards those regional and local partners, because to be upfront here, the US EPA at the national level, we are not developing any national scale regulatory framework for water reuse at present, right? So it's all at the state or local level. So we are doing research to inform them. Now, at the same time, this area of water reuse is gaining increased interest at the national level. You may have run across the Water Reuse Action Plan team within EPA, led by Sharon Napier, and that is a group that we are closely aligned with. So there is increasing national interest in this, but at this point, the regulatory actions are at the state or more local level. And then I do want to finish a little bit with talking about next steps, moving beyond this initial work we've done to think about what's over the next 5 years, because as Greg mentioned, we do this research planning for 4 to 5 year cycles. We're in the middle of that for our next 4 to 5 years. And so I'll kind of give you an idea of what we hope to focus on within this area in the near future. So, okay, there's climate change, drought, trying to look at aging infrastructure. There's a lot of needs to rethink the systems that we have. This quote is from some of the thought leaders, at least in the US, Glenn Dyer, Sybil Charvel, and others on, if you're going to rethink the water systems, you really need to put this in a historical context, because the main factors that resulted in the development of the systems we have now really no longer exist. And those are, number one, a general availability of water and other materials, including nutrients, energy, other kinds of elements, and then relative to demand, and then really a general lack of treatment technologies and the monitoring capabilities. So we didn't really need to do these advanced kind of treatment systems previously. We could kind of just use a one-way flow of kind of processing. But we also really didn't have the technology to do anything beyond the more simple approaches. And when I say simple approaches, let's be clear. I mean, the solutions of the current water infrastructure was a real solution to public health issues. So there's nothing to be ashamed of the systems we have here, but it was created for a different time and place, really the 19th century and early 20th century. So what about the 21st century? Now that we have these capabilities and the drivers have changed, the transitions in the water sector, you really can think of it as rethinking it as not just providing a service, but of a simple and separate, centralized system providing these distinct operations of drinking water, stormwater management, wastewater treatment, and really just kind of plumbing up what is already an existing city On the other hand, really thinking about the water systems as part of a circular economy where you're trying to extract useful products, including the water and other materials. And also, and what I'm going to talk about today is thinking about a hybrid approach to centralized and decentralized kinds of systems that you might use for this. And there's this term one water used. If you go on the web, you can see all these nice figures showing how we need to think holistically about water, which is one of the elements shown in the previous slide. That's true, but I think we also need to think about, given this shrinking world, that we have shrinking resources, not only do we think about a water cycle, but really think about there's tight cycles of water that we want to create, and maybe many different ones. Is there, this hybrid approach of really thinking about many tightening cycles of water, but planned holistically. And so the area that I want to talk about today is work we've done is really thinking about more in the urban environment about buildings, be they single buildings or districts of buildings, as a source of water. Not as a source of wastewater, but as a source of water. If you think about a building, there's lots of different kinds of water produced on site, right? Both the collected composite black water with other human waste. There's source separated gray water. There's also precipitation that falls on the roof and at grade, which are very distinctive in their contaminant levels, and even air conditioning condensate. So there's lots of different sources of water. And this interest in recycling at the local level in the US has been increasing interest. These show some of the examples. The Solaire in Battery Park, New York City, they've been doing this for almost 2 decades. And their driver on this was to reduce the tax on sewer flow and by recycling some of the wastewater. And they use membrane bioreactors, and they recycle that water for non-potable purposes. And in the center and in the right are some examples from out west in the US and in Portland, Oregon, and then on the far right, San Francisco, where some newer buildings have used these on-site systems to both address water limitations, but also create these green buildings and use LEED certification to really kind of improve the property value and also be demonstrated as leaders in the area. And in San Francisco in particular, they're seeing increased use of this because they have local regulation requiring any new builds over certain square footage to incorporate this type of dual plumbing, non-potable use. So as I mentioned, we started to work with this group back in 2014, so about 7 years ago now. Again, it's utilities and more local public health agencies, be it state, kind of the county level, maybe even the city level. We got together, and this map shows it from a range of the US. And I think the importance here is that when people think about water reuse, certainly it's driven by drought and the areas out west that are relatively water poor. I have great interest in water use, but there is also interest in cities back east as well in this because of what I mentioned, thinking about averting sewer flows as one example, but also really thinking even in states like Florida, where there's plenty of water, but it's not managed well, really thinking about this centralized or decentralized approach as one component of a water future. So this group really focused on, number one, local management programs are needed, but they also they felt that a real gap was water quality parameters, and monitoring. How do you tell people and how do you permit these systems to protect public health? So in response to that, we in ORD developed the research plan kind of outlined in this slide, where we looked at, try to answer 3 main questions related to, say, how do you use this variety of different water sources? How do you treat it for safe, non-potable use? And I say it's at different scales. Could be more at the smaller building size, but really it's more larger building and even districts of buildings. And these are some of the partners involved. So, but the 3 main questions, number one, how do you define acceptable treatment? So how do you define kind of the targets for the treatment systems that you're going to employ? Once you've done that, how do you implement those systems, including monitoring? And thirdly, does it really make sense to do any of this? And when I say, does it make sense to do any of it, it's really kind of related to this issue of avoiding just burden shifting. You say, okay, I'm going to recycle water locally and my building, but does it make sense? Does it have other economic and environmental impacts in terms of energy use, for example? So it really requires more of a system level assessment of these decentralized systems. And on the left, you see this is from the Water Reuse Action Plan that I mentioned, which is this EPA. It's a national federal government effort, but led by EPA, and really talking about an integrated approach to watershed management when you think about water reuse. And again, you guys, I feel like I'm preaching to the choir a little bit on that. You know this. We're a little bit late to the game on some of this at a national perspective. But my point here is that the research that we were doing on these decentralized approaches, we're really trying to look at safe and effective, how to inform safe and effective adoption of these systems. Safe in the sense of understanding the risks and developing systems to mitigate those risks, but then also effectively in terms of really thinking broadly about what the multiple impacts and co-benefits may be of this approach, and not just getting overly focused on the risk. I always, my analogy on this sometimes is that I feel like sometimes we get a little bit too focused on the seatbelt, right? We try to really focus on risk, minimize the risk, really understand all the risks, and develop better and better seatbelts, which is very important. But we also need to make sure that we keep our eyes on the road and make sure we have the car pointed in the right direction, that we are driving to a safe place in a safe way. And when I say a safe place, that safe place is some place that has sustainable approaches to water, energy, etc. So let's I'm going to talk about those of those three questions. I'm going to focus on the first one about how to define acceptable treatment, and the third, the system level analysis. That second question about monitoring, we had some active research in that, but over the last really 2 years now, that's all those resources and effort have really been diverted to the SARS wastewater surveillance effort. So we really have not been able to progress much there, but that will be a continued effort in the future work. So we'll talk about these two questions. So in terms of how do you define acceptable treatment, in this case, you have different quality of alternative source waters. You also may have different scaling effects for decentralized systems. You think about fewer people. So there may be a larger percentage of the time where people are not infected with the pathogens that you're worried about removing. But then when someone is, there may be less dilution of that wastewater from other sources that you'd have in a centralized system. So trying to understand what the scaling effects is important, and then really understanding then fit for purpose use. How are you going to use that water? In this case, we've mainly focus on non-food irrigation, landscape irrigation in the urban environment, as well as toilet flushing and clothes washing inside homes. And we focus mainly on microbial risk assessment because for non-potable use, the chemical risks are not as large a concern. You really are interested in the infectious risk and kind of acute risk from that. So from a microbial, I don't know, I assume most of you are familiar with this general QMRA approach. Generally, you want to understand what the density of pathogens are in these different source waters. That's a critical factor here and one of the gaps in a lot of the understanding for these different waters. But you also want to know what exposure volume for a particular end use you're going to have. You need to understand the dose response, and there's some data on that, but it's not complete, and there's always room for discussion about improvements in that. But then once you have estimates of those things, you can predict what the risk is of a given reuse of a given water. And then you define what your tolerable risk is, and the difference then is what your reduction target is. In microbial terms, it's usually log reduction because of the numbers. So this log reduction target gets you from predicted risk to tolerable risk. Now, tolerable risk, I'll talk a little bit about that. We tend to use 10 to the minus fourth or 10 to the minus second infections per person per year. So it's an infection number of infections per year. But I'm going to talk a little bit as part of this. We've started to get into antimicrobial resistant infections, which can have deeper impacts. or broader impacts on your health. So infection may not be as good a predictor, but for most part, we look at these two estimates of 10 to the minus 4, 10 to the minus 2 as different markers for tolerable risk. But you know, tolerable risk, what is tolerable? That's kind of a regulatory issue and certainly a regulatory decision point. And so the general idea is that, you know, we were looking at a range of source waters for a range of end uses, and you end up with this matrix of log reduction to targets to define fit for purpose reuse. Pretty straightforward, pretty simple, and I'm sure I apologize if I'm presenting oversimplified issues to you. The results of that work, getting back to this Blue Ribbon Commission, we generated a number of estimates for both domestic wastewater, black water, gray water, storm water, roof runoff water, and for their unrestricted irrigation or indoor use, what the log reduction targets are, again, for 10 to the minus 4th, 10 to the minus 2, for the different groups of representative pathogens. So viruses, protozoa, and bacteria are the way the QMRA is structured, so that you really want to have log reduction targets for each one of those groupings. And then you pick the pathogen of most concern typically with the viruses. We use norovirus because of its high infectivity and low dose response. So, but you know, you can vary the type, but it's important that we have this grouping built into the quantitative microbial risk assessment, because as we move into chemical risk assessment, which is next steps, having some similar kinds of groupings of chemicals is going to be very critical, I think, to developing an effective framework there. So, aside from that background here, these are the numbers that this collaboration with this Blue Ribbon Commission, these data have been generated. We developed a series of peer-reviewed publications. This group adopted those and came out with this final report, this risk-based framework. It's like, here's what, if you're going to develop these non-potable reuse systems, here's the targets that you should try to use for permitting. And now these data have then been transformed into state-level legislation for non-potable reuse in the states of California, Colorado, and Washington. And I think there will be more over the coming years. So let's dig a little bit into the details of you know, how some of these numbers are generated. As I mentioned, the pathogen density characterization. Now, understanding what your contaminant level is, i.e. pathogen levels of concern, and we're not talking about fecal contaminants here. I mean, fecal indicators. We're talking about what the pathogen levels are in that wastewater. And so for wastewater and gray water, especially at these small scales, we went out to the literature first to get data on that. But at scales of that I mentioned, this scaling issue with decentralized systems, there really was not a lot of available information, not sufficient to generate a robust distribution that you would feed into these QMRA models. So but there was a lot of estimates, a lot of data on fecal loads based on fecal indicators. So we developed a method that linked the fecal contamination based on the fecal indicator levels with kind of a shedding rate based on what we think the frequency of infection is in populations of different sizes. So it can be scaled to different size populations. That then ends up with estimates of pathogen concentrations in the water. So we've developed those models, and we then went out to test those to kind of validate how well they predict the levels of pathogens in 3 different decentralized systems. These were all about 500 to 1,000 people, occupants in the buildings. 2 were source separated gray water, 1 was a combined wastewater. And we looked at norovirus and adenovirus in particular. Viral risks tend to drive a lot of the risk, so we focused on virus to begin with. And so this data, what you see here is this is measured values. This is from our partners in San Francisco, from their building where they have a reuse system in place at the Public Utility Commission building. The dotted lines and circles, those are the measured values for norovirus using qPCR and digital droplet PCR, which are two forms of estimating the frequency or occurrence of the gene specific to norovirus. So this is not a viable virus estimate. This is an estimate of what the gene copies present are. And now that's another key issue for the understanding risk, because you're really worried about infectious virus, not the mere presence of the gene. But in this case, the thought is that it's fresh wastewater, right, where this is collected with it before any treatment. So we're using these as proxies for infectious virus. But anyway, so here's the levels that we measured over the course of about 7-8 months in that building. The value here, this is the estimate, this is the mean estimate, and then there's the 75th and 95th ranges from our modeled predictions. So you can see that the modeled predictions do pretty well. They fit both the kind of the rough average and also the range there within the 75%. So that's the black water. The gray water, in two systems there, we looked at adenovirus and norovirus in those systems, and then we also simulated it for those populations. And so you can see the measured levels are a little bit lower. So this table over here kind of shows what we quantified, and a lot of times we got a lot of zeros in gray water because it's a lower concentration. There's some detection issues here. Even with some of these, we see improved detection with digital droplet PCR, and that's why we use digital droplet, because it's more accurate at low concentrations. But bottom line is that it's lower than the simulation levels. So our simulations may be over predicting concentration, so they're probably maybe a little bit on the conservative side, but still within a reasonable range. So another thing to consider with this on-site reuse is potential for cross-connection issues. And there's a couple different cross-connections that you could be concerned with. That's the reclaimed water that you're using to the potable line that's in the system, right? So you've taken the raw wastewater, gray water, treated it, and then there's some kind of cross connection with some dilution into your potable water in the building. The other cross connection of concern is the raw wastewater itself, being some cross connection between that and the already treated non-potable water for ingestion. And so we've done some work on modeling both the duration, the percentage of the population exposed, and the dilution. And if I've got all the papers listed in one of the coming slides. So if you want to really dig down into this, you can see the analyses that we've done. But we've included an ingestion safety factor for indoor use. And that assumes a relatively low level, short duration, relatively dilute cross connection that can happen at some frequency. So we build that into the model based on some of these estimates. But I would say these are all modeled estimates. There's a lot of lack of data out there for real world cross connection in these buildings. But then when you take that cross connection, right, and you have this kind of safety factor built in, this graph, what it shows is some other work that we did is we kind of try to look at what is the different predicted LRTs across a range of expected ingestion from different alternative end uses. So, for example, showering, you might expect to have a higher ingestion volume of the treated water, just because as you're showering, you can swallow some. For indoor use, like toilet flushing, clothes washing, it's just really relatively low. It's from droplets that are produced, vapor, when you flush toilets or when you're washing. So relatively lower level. Then there was some that there really was there's been some studies on showering and toilet flushing exposures. For decorative fountains, for example, or irrigation, we also had, there's some data but less known. But we really want to understand for these exposures where there's relatively low ingestion, what we find is that when you build that safety factor of the expected level of cross connection into your systems, When you get to these low-level exposures, your LRTs, your predicted LRTs, remain kind of flat. That's because the main exposure then is from not the ongoing predicted daily risk from the use, but from this intermittent risk from cross-connection risk. So what it means is that even if you don't have a high confidence in some of the specific exposure volumes that you get from some of these alternative indoor uses, the models that we have and the LRTs based on those models, because they include this safety factor, probably address those, and you're all right. Now, what I've talked about so far is wastewater, gray water. I mean, there are other source waters or alternative source waters of interest where there is also gaps in our understanding of what the contaminant loads are in those. For example, roof runoff, there's very limited data. There's fairly good data from Australia. They've collected some reasonable data from rainwater in Australia, but that's Australia. They have and the drivers for roof contamination are mainly animal zoonotic, animal use of the roof and zoonotic risk from that. So we in our initial models that I showed for the LRTs, we kind of estimated what there were data from what the bacterial contamination was in Gulf feces, and so we really modeled from there. But we knew that there was a gap in data, so we collaborated with Colorado State University, who sampled at sites across the U.S. Particularly, we were focused on Tucson, Denver, Baltimore, and Miami, where they collected fresh rainwater samples off the roof from those 4 cities. We had about 80 samples. Still not enough. We'd love to have hundreds of samples, but this was more than before, and we looked at 13 rain events. And then in this case, we looked at the Salmonella and Campylobacter occurrence. So these are heat maps. So for those cities, you can see when we detected it and what the concentration was by the intensity of the color. So you can see that we saw mostly Salmonella occurrence, not a whole lot of Campylobacter, but we did see Giardia in some of the samples, and this would be from mammalian use, rodents, squirrels, in the case in the U.S. a lot. And so that was again, these were relatively lower levels than our models had predicted originally, but we had not even modeled Giardia because there was no data on that. So this suggests that we really need to include LRT for protozoans in our roof collected rainwater estimates, but we still have fairly limited data for that. Another source of water that we're really interested in using is stormwater, right? So, and if you noticed in that original slide, and not to make your head spin, but if you go back to this slide here, we estimated stormwater as a 10 to the minus 1 dilution or 10 to the minus 3rd dilution. That's of untreated wastewater. Because if you look at the data that's out there for collected stormwater in urban environments, that's the range of pathogen levels that you tend to see in stormwater. Even though the data is limited, it suggests that stormwater is relatively highly contaminated due to what we think is largely poor infrastructure, leaking, exfiltration from the existing infrastructure. But the data was limited, and so we estimated stormwater either as basically a very contaminated or 10 to the minus 1, 10-fold dilution of wastewater, or less contaminated 10 to the minus 3 dilution to generate these terms. When we've rolled this out, that's obviously been a concern. People really want to know whether they need to treat it to that great a degree of difference, right? How clean is my stormwater, basically? And so that's a real issue moving forward, because this dilution of municipal wastewater, it does reflect that it's highly variable, the measurements that we have, and it kind of addresses that there's very limited data to base anything else on. So it does provide flexibility, but it's ambiguous of what's right for a particular project. So what we're moving towards in the future, in collaboration again with Colorado State University, who has a Water Research Foundation grant on this, is to try to understand an integrated assessment of your site, including physical characteristics, the hydrology, the contamination sources, and also the infrastructure condition, and then probably also involving some validation measurements, if not of the pathogens themselves, of human-specific fecal indicators that would drive those models, to then kind of bin your stormwater into higher or lower quality to define the treatment requirements. Just a couple other things real quick on the risk side. There are existing permitting systems in the U.S. This is not the National Science Foundation, but it's the National Sanitation Foundation. They have developed a process for permitting and validating on-site systems, but it's based on measuring the removal of fecal indicators. And again, the fecal indicators are not the things that get you sick, right? All the models that I've mentioned are based on pathogen levels. So what we did is we went in and did a QMRA on the systems that NSF has validated based on the indicator-based standards that they use, right? Because what they require is that you challenge your system with certain amount of sewage and you measure fecal indicators in and out. But what we found is that for certain types of wastewater, residential gray water, for example, the estimated risk is 10 to the minus 6. So it would be below it would be above the acceptable or below the acceptable level, right? It would be acceptable levels of risk by 2 orders of magnitude. But those systems can be used for other sources, including larger scale residential systems and also business gray water, which is less diluted by showering, et cetera. It's more hand washing water. And then finally, wastewater, which has a higher fecal load. Those then are not protective of public health. And I think what this suggest is, and we've talked to NSF about this, is that we really like that their performance-based approval system. That is a very good thing, but we need to make sure that we push the field into measuring the right targets in that performance testing, not bacterial indicators, but the pathogens themselves or surrogates for those pathogens. And then finally, I've been talking about, these are all risks due to enteric exposures, right? So risk of virus, protozoan, even bacteria that cause enteric infection, so swallowing, ingestion. But there are also, in some of these reuse applications, there's skin exposures, which we also need to understand that risk. So we have started to work on Staph aureus, both susceptible, antibiotic susceptible Staph aureus, as well as a multi-drug resistant Staph aureus or MRSA, and try to understand what the risk from exposure from the skin to those are. So, and we've adapted the QMRA to try to accommodate for the antimicrobial resistance. This is a little bit more complex because you're also interested in exchange of the resistance during the potential treatment. So it's not just what's in the original water generated, but also could change during the treatment. It also it's a little more complex because you can be colonized by the resistant form, and then it can then subsequently cause clinical disease. And then the other really key factor with MRSA versus susceptible Staph aureus is the severity of the disease. And this gets back to the point that I mentioned earlier about all the risk models I mentioned today were driven by the pathogen occurrence, the infection level. But you can also convert to disability adjusted life years, which accommodates for both the frequency of infection and the basically the impacts of that infection, right? How many life years, including death, does that disease cause? And clearly, a resistant Staph aureus is much bigger impact disease than susceptible. So we've begun to look at those things, and there's a paper outlining our initial work on this, which in essence shows that for number one, it shows a framework for how you begin to look at antimicrobial resistance in this risk framework. But then also it shows that if you just look at infections, our current targets are fine. But if you want to start thinking about disability adjusted life years, which is something that the WHO use, but it's not as widely applied in the US. But that's something you really need to try to factor in when you're thinking about AMR. So this first part of the talk, let me just summarize what I've just said. I mean, I think we've developed these risk-based targets for enteric pathogens, and they've been adopted by multiple states. We've modeled the predictions of pathogen levels for gray water and black water, and they seem relatively consistent with the more recent pathogen characterization data that we performed. This built-in safety factor for short-term, low-magnitude cross-connection events, we built that in, and that also allows treatment targets to be extended to other uses with relatively small exposure volume. So you don't need to build new LRTs, just use the existing ones. There's greatest uncertainty probably in the pathogen characterization for roof collected rainwater and stormwater. We've got some improved national scale data for rainwater. It's relatively low risk, but it does raise this issue of protozoan risk and trying to deal with that in the treatment. And then finally, we do need these new models to look at high and lower levels, to bin out stormwater into high and low levels of contamination. And again, as I said, the treatment systems validated by fecal indicators, and this whole general idea of using fecal indicators as the endpoint, we're really trying to move away from that and really focus on this pathogen-based, risk-based approach. And then again, we said we've started to do work with QRA. So that's the first part. I'm going to see how I'm doing on time. This is just a review of the literature on this. So I'm going to, Sally, I've sent this slide to you. We can share this with everybody here. So if you can see any of these, and if you have any questions about any of this work in more details, feel free to reach out to me. Michael Jane, who is co-author in all this work too. He's the real in-house expert. Mary Shane is someone who works on contract with us, both experts in this area. So let's kind of turn to this.
Jay?
Go ahead.
Jay?
Yeah.
Just a quick question. You've talked basically about microbial QRA,
Yep.
QRMA. Do you see chemical risk and some of the risks for CECs, PFAS, PFOA, those kinds of things influencing your priorities in terms of what sort of research you might be doing on not just microbial but chemical type risks?
Yeah, definitely. That's a very good question. And I was going to touch on it briefly at the very end in terms of next steps, but that's, we focused on the microbial risk because these were for non-potable applications. But as you move to either chemical or either alternative source waters that are mainly chemical risk driven, one example that is produced water, right? Water that's produced from fracking operations, and there's interest in trying to reuse that. There's no microbial risk associated with that, but there's chemical risk, so trying to understand the risk there. Plus, as you move to more potable end uses, which could include direct potable use, but also use for agricultural operations, and that's where we really need to build in new frameworks for looking at chemical risk, and that's a major area of our next 4 or 5 years. And I think the key to that is really thinking about new approaches that move away from try to think about all the chemicals individually, but really thinking about a broader framework that groups those contaminants into different relevant classes, much like we do on the microbial side here, right? Viruses, protozoan, bacteria. Can we think about doing something similar to the chemical risk side to both define treatment requirements and then also think about what target chemicals or even bioassays that we may want to use to understand the level of removal in those wastewaters. So yeah, that's a key issue moving forward as we move to more potable use applications. Other questions on this part before I move on?
Yes, please. If I could ask, please, is there a need for licensing of treatment facilities at homes, and do they have to make regular laboratory checks I would just say that we had a big question about centralized and decentralized treatment regarding especially gray water in Israel about 15 years ago. And we decided not to allow gray water use in homes, especially because when you take in factor that, let's say in United States, if potentially each house would have it, we're talking about hundreds and thousands of treatment plants with a potential of, as you said, connection to water, and that would be. And so, how do you deal with the risk assessment regarding so many treatment plants?
Right. Yeah, and I think the issue there is not as much, defining what the treatment level needs to be for it, but it's more about how do you ensure that you're getting kind of continued performance, right? That there's necessary O&M, operation and maintenance done, and you're really meeting what the original prediction. Our collaborators on this, like I mentioned, the state and local partners, they're very interested and really focus more on the building scale kind of systems where there would be a kind of an operator, and that sometimes, in some cases, some of the utilities are interested in taking that on as part of their role, but there also could be private companies that are responsible for operating that system. And for the Solaire example that I mentioned in New York City, there's a company that has been operating that system since its inception. So in that case, there's more of a focus put on a professional operator, and that they then have to report out to the permitting agency, which is usually the local authorities, on some routine basis. But I agree, when you move to the individual house level, there's a lot of concern about public health there just because of poor operation of those systems. So most all of our focus in this group has been, like, if you look at this slide, not on this household level, but large building and district kind of scale things. But that said, there's a push by a number of people globally, and this is some corporate entities in particular that are pushing for, like, developing a 50-liter home challenge, right? Like a house that only would lose 50 liters, and so that there really would be gray water reuse at a building, at an individual household level. And that's, I don't see our partners interested in adapting. Again, the city's adapting that. So similar to your case, I think they want to stay focused on larger buildings as opposed to individual households. But it's still out there. People are talking about these individual household systems. In fact,
So-
there's some experts that talk about this extreme decentralization, where if you really want to make progress, that's what you need to do. Really, they use the cell phones as the example. Turn it over to, that's where you're really going to get increased novelty and new technology. It's going to drive the success of these kind of systems by turning it over to more personalized, individual-based systems. But that's one more step in the change. Sorry to interrupt your question.
No, you don't have to license the treatment, and you don't have to. It's not compulsory to make lab checks.
Well, that NSF 350, that is a system to kind of approve, so you can but at the requirements that you're mentioning would be at the local level. My understanding from working with San Francisco and Denver, city of Denver, for example, they're requiring that there's a engineered design for your system that meets these log reduction targets. And as long as you've documented that it meets those targets and it's signed off by a professional engineer, that it's approved for construction. And then in terms of there will be I think the need for some check of the system, right? So, and that's going to vary. I forget in California, in San Francisco, I don't know what their frequency is. I think it might be weekly or monthly. But part of the challenge there too is that the models have gotten ahead of the methods because they're requiring checks of the system, but it's usually fecal indicator data, which I kind of emphasize that that's not what you really want to monitor. You want to understand pathogen removal. So that document that I showed, this one here, this report emphasizes that you validate that the system performs in terms of removal of those pathogens as designed, but the monitoring really should be not on fecal indicator organisms because it doesn't really tell you about the risk. It's more about parameters. For example, if you're using a membrane bioreactor, which is very commonly used, that you ensure that you have online measurement of transmembrane pressure and that you have continuous suitable operation and maintenance of a transmembrane pressure, and then also that you have some measurement of your oxidation. Maybe it depending on what type of oxidation it is, you have a certain UV fluence or a certain disinfection residual in place. And so that the state of California, they have just recently passed, they implemented this risk-based framework. Their legislation is passed. Their regulations are coming out probably at the end of this year, and they're going to be very focused on they're going to allow you to do this, but the types of systems that you're going to use are going to be very limited to membrane bioreactors plus advanced oxidation. And so I would expect that their requirements are going to be around monitoring the performance of those systems, not a routine water quality fecal indicator measurement.
Jay, don't most of those things require a professional licensed operator?
Well, I think what they're requiring now, that it requires a professional engineer to sign off. But one of the things that this National Blue Ribbon Commission has done is they've developed a training package for operators of these kinds of systems, because it's different, right? It's kind of drinking water and it's kind of wastewater systems. So they've developed a training module for these kind of folks, and they're trying to work with some of the accrediting institutions in the U.S. to kind of push that forward, so to make sure that there's not only are there accredited people, but they are appropriately trained in this kind of new approach.
Okay, Nomi, I think you have a question. Take yourself off mute.
Yes, I'll take myself off mute. Thank you. It's kind of a question. Such a lot of research about the risk in doing this, and you're getting to the point where buildings, neighborhoods, or whatever are being allowed to do this. In Israel, we haven't got that far. And I often wonder if research is being done on the economic and environmental benefit of doing it, as well as the health risk of doing it.
Well, you are that's a great setup for what the next portion of the talk. So I would suggest, especially given time, give me I guess we have probably about 15 minutes more, right, Sally?
Yep. Mm-hmm. Yeah.
I'll go through the remaining slides, and it really speaks to that question of, does this make sense to do, right, from an economic and kind of environmental impact perspective. And so we've done
one more question.
Okay, sure.
One more question.
Yep.
Laura?
Hi, thank you so much, Jay. I appreciate your lecture. I have two questions for you that are related to this topic. As you know, we use a significant amount of treated wastewater and also harvest stormwater as a growing and almost a primary source now of agricultural irrigation. And in my own research, I have measured concentrations of pesticides and pharmaceuticals. So my first question is, we've now demonstrated that our irrigation source is contaminated with these pharmaceuticals and pesticides. And I'm wondering if there's any standards, or have you begun to look at developing standards or risk impacts, health impacts, both to humans and to some of our target ecological indicators?
That, from the reuse perspective, no. I mean, that's, again, as I mentioned, EPA isn't regulating any of this, right? But I mean, our collaboration here has been largely on these kind of urban applications, but this issue of broadening the reuse to think about more of the watershed level, i.e. really thinking about agricultural applications, is, I think that is one of the key next steps. So you guys are probably definitely ahead of us there, because you've implemented these systems and now you generate the data. We'd be really interested in working with you and understanding your data, because I think we need to move in that direction. But
okay,
simple answer to your question is we really haven't done a whole lot on that yet.
Okay, great. Well, we're just kind of beginning some of that ourselves, and we'd be very happy to collaborate as we advance together. This is a related question, but it's connected to what you were just saying about expanding the framework. And for example, what we're finding is a cocktail of these mixtures. So I've identified in 1 liter glass bottles in one location, approximately 35 to 40 different pesticides. So it's, we're trying to do this risk assessment and evaluation of the impacts for the ecotoxicological analysis. Most of the world is based on looking at the risk impacts from a single compound. Right. And it looks like you're working on this framework, but how do you see that moving forward? And do you have some new approaches to looking at this cumulative cocktail effect?
Yeah, I think the direction that we're trying to work in is in collaboration. One of the other programs that Greg mentioned to begin with was the chemical safety and safe and sustainable chemicals program, which is, and then we have our computational toxicology center. And so they use a lot of new approach methods, both in terms of high throughput methods to evaluate toxicity of a wide range of target compounds, but then also really thinking about adverse outcome pathways and trying to orient that large number into maybe certain adverse outcome pathways of interest, and then targeting bioassays or effects-based bioassays on those different adverse outcome pathways of interest. And therefore, then, both the monitoring approach for looking at the quality of the water, and even the risk assessment approach for determining the level of treatment might be built around more of those effects-based bioassays than the strict measurement of the range of chemicals. Because no doubt that we can look at that range of chemicals, but sometimes it's overwhelming, right? So how do you streamline that into an effective framework for both defining and monitoring treatment performance? So that's kind of that's the path that we're starting to go down. But like I mentioned, we're just early in the game on that, and sounds like you all would be very good partners in that.
Okay,
thanks,
we'd love
Jay.
to. Sorry, I'll just finish with, we'd love to join you in that effort. And if you could provide the contact that you just mentioned, who's kind of leading that team, I'd love to kind of reach out and maybe we could, begin working on it, because it's kind of timely that we're in that now. So thank you so much for taking the questions and for your presentation.
You're welcome.
Okay, we don't want to interrupt you any further. Keep going, Jay.
No worries. I think that's a good natural point to talk about the risk, because this is a different topic. So the other side of this is really trying to think about what the impacts of this type of system are, right? And what we've done is use a life cycle approach to look at the relative cost and impacts of these different systems. The first thing you have to do is lay out the entire system, do an inventory of all the, in essence, cost and impacts of individual elements. So what we're talking about here is non-potable recycling of water at a building, where normally you'd have centralized water treatment, centralized delivery of water, wastewater generated, and then that taken out to centralized wastewater treatment. And for the models that we're looking at, it's been some portion of that is diverted through non-potable demand, and we've looked at the relative impacts of the collection. We've looked at actual thermal recovery of the energy that's in that water, converting that thermal energy into electrical energy, other pretreatment needed, and then we've looked at a range of target treatment systems ranging from aerobic MBRs, which are the kind of the most commonly used right now. We've also looked at anaerobic MBRs as an emerging technology because it also has lower energy costs for operation and also biogas generation as an anaerobic process, so it might have benefits. And then we've also looked at kind of recirculating vertical flow wetlands as one option for a plant-based system. And then you have disinfection, some storage, and distribution. So the point is you got to look at the whole system and do the life cycle, kind of a balance of that. So, and this just, kind of looks at, the different steps, to get in the weeds here. I mean, anaerobic MBRs, when you treat it with anaerobic MBRs, they have, there's reduced nitrogen. The nitrogen isn't oxidized during the process like biological aerobic treatment. So we have to have additional systems to remove that ammonia before it goes into be disinfection, or it's just going to consume all of your disinfectant. So you really need to think about all these multiple steps. So anaerobic membrane bioreactors may seem good, but there's still some extra post-treatment needed. So the bottom line is that what you can then look at is you get this cumulative kind of picture of an individual technology. So in this case, we're looking at global climate change potential. So the kilogram CO2 produced per year in a large building from a given amount of recycling of water. And I know this slide's kind of busy, but what you're looking at here is things above zero are the cost of the system, typically the operation of the system. Then below zero are the avoided costs. So this blue bar represents the avoided cost of supplying that drinking water to the site. So both the cost of treating and delivering that water. And then these striped bars here are if you are generating energy locally, either by recovering the thermal energy, and that's this case here. This is an aerobic MBR using thermal recovery. This is the aerobic MBR without the thermal recovery. This is the anaerobic MBR. And so the bar here is not thermal recovery energy, but it's the biogas that's generated. And we've looked at anaerobic MBRs two different ways, kind of traditional operation, also kind of with a reduced intermittent aeration to reduce operating cost. And ANBRs are still in development, so we kind of looked at more of the literature values of, and this is a proposed approach for improving efficiency. And then finally, this is the recirculating vertical flow wetland. Bottom line here is that the net benefit is this red. So if the net benefit is actually positive, you have a negative score here. So it actually it's a net benefit reducer of global warming potential. So you can see that a lot of the systems, particularly the aerobic MBRs, which have the thermal recovery, even the vertical flow wetlands, even the anaerobic MBR with this kind of intermittent aeration, they are net benefits from a global warming potential. And then from a cost, we also estimate that. And same kind of graph. I won't go into detail on that, but that's the kind of the overall system cost of that. So, what we found is that, the displacement of drinking water and associated impacts, you really need to factor that in. But when you do, these systems at a local level can be a net benefit. The aerobic MBRs seem like the most economical approach, but partly that may be driven just because they have been established. We know more about these. We have more experience with them. They've been fine-tuned more because of their application. Anaerobic MBRs, recirculating vertical flow wetlands they have some additional pre or post-treatment processes which increase their cost, but maybe that's a temporary bias because we just need more experience with these new systems. And there is some cost of additional piping network that drives the source separated gray water system. So there is some cost benefit for just recycling the overall combined wastewater. So we also then, just to finish up here, I wanted to talk about we've taken this kind of to a broader scale. That was like a targeted study at a building using different systems for applied to San Francisco, so our partners out there. But we really wanted to build a tool that anybody in the US could use. And we call this the newer non-potable environmental and economic water reuse calculator. So what this does is that, and when you get the slides, you can play around with this if you want to. It's for the US. But it's intended for developers so they can calculate as a kind of a screening tool what the cost would be for different designs. And we're interested in this could be the Green Building Council, because we factor in these things like global warming potential, it may be interested for them potentially eventually for their LEED certification. So the newer tool allows you put in a location, your 5-digit code, your zip code, you characterize the building size, what source water you want to use, what the end use for that water is. The first thing it does, it kind of, for a given zip code, it estimates what your water availability is for that. Now, some of the waters are going to be location specific, right? Rainwater off the roof, atmospheric AC condensate are going to be dependent on the environment of the zip code. Others are going to be more dependent on how many people in the building, right? They're going to be climate independent, wastewater, gray water generation. It also calculates in the US, we have a wide range of what the source of electricity is, and therefore what the global warming impacts of those different grids are, right? So that's an important factor to consider. So if you look at the availability, again, this looks at the rainwater availability and atmospheric or AC condensate availability in the US. In the United States, Southeast, very warm, very humid. So there's both a lot of rainfall and a lot of AC condensate produced. There's also a lot of rainfall on the West Coast. But in most of the places, there's relatively little. And if you express it as a percent of the non-potable demand that the building needs, you can see that in most places of the country, it's going to be less than 5 or 10% of the water. So these sources of water, other than maybe in the Southeast, where you combine between rainwater, atmospheric, or AC condensate, maybe you can meet 30 to 40% of the non-potable demand of the building. It really is not going to meet a lot. Now, the gray water, black water, in all sites, it meets 100%. And so the other thing we did with this newer tool is we actually went in and looked at different simulation sets. We kind of ran this newer tool for different size buildings. And also, so one was we kind of had a standard size large building. And then the next one, we kind of looked at a random generator of a whole range of building sizes to generate this kind of data. And this gets to, when you talk about global warming potential, it depends on where you're at in the US. If you're in an area where it has a very carbon intensive energy grid, like this portion of the US, if you're treating water, be it gray water or wastewater with an MBR, there's a very high cost of that. And actually it's a net consumer of energy. But if you live in areas with a greener, you're a little bit better off. But then this is with thermal recovery. And with thermal recovery, if you're actually using, especially offsetting electricity, well, you really can get into anything in the green levels are net beneficial global warming, right? So they actually are reducing global warming potential. So again, in these areas with a high energy grid, if you're trying to recover thermal energy and make it into electrical energy, boy, you really, so this is complicated, but it depends. So it tells you really what the importance of where you're at based on that electrical grid. And then these data, what we've done here is looked at global warming potential across a size of our system. So remember, we did this randomly selected different size buildings in different places across the country. And what this is for rainwater harvesting is blue, AC condensate harvesting is green. The global warming potential tends to be very high for these small scale systems because you're building this whole system to put in place for relatively small volume of water recovery. So the global warming potential tends to be pretty high associated with the actual construction of that system. Now, as you get into larger systems, they again get into the net zero levels. If you're in, so this would be in some of those sites in the U.S. like Florida, other parts of the Southeast where it could be beneficial. For the gray water and wastewater, and this is no thermal recovery. We're not looking at thermal recovery. They tend to be, they're slightly higher. There's more volume, but this is associated with the higher treatment cost that you have to do with these than with, the rainwater and AC condensate are relatively clean, so low treatment cost. Here you have higher treatment cost. But again, this graph shows that depending on whether you have thermal recovery, not no, but thermal recovery, that's if you're in areas with very high E grids, boy, you can really start to get into the net, the global warming reduction area, right? So, and then these are the costs, the cost of these systems. Again, as you get larger, the costs come down. And if you divide the net present value by gallon by the potable water cost in that area. So in this case, when you divide by the potable water cost at that site, anything about 1, so if it's below 1, it's actually cheaper water that you're producing. So in most cases, it's more expensive water than you're producing from than the potable water. But for some of these large systems, where you're recycling gray water and wastewater, and depending where you have relatively high drinking water costs, which are increasing all the time, you actually the water is actually cheaper that you're producing locally. So complicated picture, not straightforward answers, but things that you need to look at both geographically, explicitly. So just in summary, most areas of the country, rainwater AC condensate provide less than 10%. Now, where it's available, these water sources provide an environmentally beneficial, because there's low treatment costs, but relatively costly option for reuse because it's a relatively small amount. For wastewater and gray water, they provide 100% of demand. Those energy demands for treatment lead to environmental impacts, and it's really driven by the carbon intensity of your energy grid there. But it can be a cost-effective source, especially where drinking water costs are high. So we're really really hoping that people can use this tool to develop these kind of localized cost-benefit assessments. Again, here's the literature for this kind of work. And just to close out, I think I've got a couple minutes. We've talked about this. We want to really move into estimating chemical risk. We want to improve prediction of microbial risk with stormwater as well as chemical risk. And we want to expand the implementation. We want to do we really kind of look at in doing this work on quantifying and monitoring tree performance, work that we wanted to do early. It's that second question. We think we need to revisit that. And then secondly, we want to expand the boundaries of water reuse, really look at more integrated water management, much like you all have. Really think about not just this urban use, but also agricultural use, more integrated from a watershed perspective. All right, so sorry, we're almost out of time, but I guess we got a couple minutes. I do have a 10:30 I have to get to.
Yeah. All right. Thank you very much, Jay. I would just invite if there are any questions, any additional questions for Dr. Garland. Go ahead, Dave.
If I may just add to the question about, the situation in Israel about gray water, we have quite a different situation because in Israel, every drop of water is already used twice. Once in the household and second in agriculture. The only time you would use it 3 times is if you flush toilets. And we decided this won't be a great change. So today it is allowed only in businesses which have to have a license, and we check the treatment plants, and they have to send samples to laboratory to show that it all works okay. So generally in households, we do not allow it.
Yeah, I think that makes a lot of sense, and it speaks to maybe some of the differences in the scale, right, geographical scale between our countries, right? I mean, so as we move to thinking about expanding reuse to a watershed scale, there may be certain watersheds where the Israeli approach of really focusing on reuse for ag makes sense, but in others where it doesn't because of the transportation cost of moving that water, right, from larger distances, for example. So that's a really good point, and just speaks to, like I said, differences in geographic scale. Well, I apologize. I'm sorry I took a little bit too much time. Hopefully I answered some questions in the middle there.
Yeah.
Feel free to reach out, but I do have to kind of get on to my next meeting. I apologize.
All right. Thank you very much, Jay, for
You're
the
welcome.
excellent
Take care.
remarks. Wow. So now I know we are at time, and I just wanted to thank you all again, all of the participants from the Ministry of Environmental Protection, Health and Agriculture, Mekarot, and so many others that have made the time to attend these webinars. We really hope that they have been useful as information sharing. I also wanted to thank our colleagues in the U.S. Embassy in Israel for their cooperation and their support. As we said at the start of the webinar series, water quality management in the U.S. is very complicated. It's a lot. And it is not possible to explain all of the different dimensions in a 5-part webinar series, obviously. But we hope that we can continue to provide additional information, collaboration, and expand our partnerships. So I want to thank David and Phyllis for their fantastic leadership in all of this. And I know that there was a lot of work that was done behind the scenes. So I so appreciate that. And from the EPA side, I want to thank Teresa Kuklinski from our Office of International and Tribal Affairs, and my colleague Maria Lopez Carbo from the Office of Wastewater Management Water Permits Division. Thank you all so much. And I hope each of you has a wonderful 2022 as we look forward to that. So more to come. All right. Thank you.
Thank
you
you
all.
very much.
Thanks a million,
Thank
Sally.
Uh-huh.
Thank you so much.
Uh-huh. Thank you.